next one is the method for Magdalena Plotczyk, the role of hair follicles in dermal remodeling and scar reduction.
Thank you for the introduction. It wasn't an easy job to pronounce my surname. I would like to thank the organizing committee for selecting my abstract and giving me this opportunity to present my research today. And I would like to thank the EHRS for awarding me the travel grant to attend the Congress. So I'm a PhD candidate in the lab of Dr. Claire Higgins, just starting my second year of the project, and I'm going to tell you about my work looking at the role of hair follicles in human dermal remodeling and scar reduction. So the reason I got interested in this topic is because researchers and clinicians have noted for years that hair-being areas tend to heal more quickly than areas lacking hair follicles. And this wound healing capacity of a hair follicle was recently supported by a clinical study in which punch grafts from hairy scalp and non-hairy abdominal skin were transplanted into venous leg ulcers. And the ulcer healing was measured as the average area reduction. And it was demonstrated to be significantly faster in the hair follicle group. And after finding out these striking results, I decided to undertake a project looking at the mechanisms by which a hair follicle can affect the healing. So what we know already is that the epidermal stem cells can migrate out of a hair follicle in order to promote wound epithelialization. However, it is not so clear how the dermal follicular populations, dermal sheath and dermal papilla cells, can contribute to wound repair and remodeling. What we also know is that during the hair follicle cycle, the interfollicular dermis is remodeling, and the changes include formation and remodeling of extracellular matrix fibers and local increase in vascularization during the anagen phase. And this is actually the opposite to what we can see in unremodeled scar tissue. So this is why in my project I propose that these factors are the possible mechanisms by which a hair follicle can induce dermal remodeling. So in order to answer the question whether hair follicles could promote wound healing and reduce scarring by affecting dermal remodeling, we've established a clinical study in a collaboration with Dr. Francesco Jimenez. So follicular units will be transplanted into established stretch scars on the occipital scalp, and 4 biopsies will be taken at different time points before and after the transplantation. And this will allow us to see the progressive dermal remodeling as the direct result of hair grafting. So far, we recruited 2 patients to this study, and a full set of biopsies was collected from one of them. But before the study is completed, I want to first characterize the unremodeled scar and remodeled healthy dermis in order to establish a baseline for our patient study. So despite many people looking at the scar tissue, the published studies were mostly about pathological scarring such as keloid or hypertrophic scarring. But in contrast, I will be looking at samples including stretch scars that resulted from an applied mechanical tension to the wound healing area. And the 2 interesting properties that were previously reported to differ between scars and healthy tissue are the composition and alignment of extracellular matrix fibers and the mechanics of the skin. So in my study, I used second harmonic generation imaging, which is a very attractive alternative to standard fluorescent-based microscopy. And this is because of a high tissue penetration, lack of phototoxicity, and no need for staining, which results in this high signal-to-noise ratio. So I used this multiphoton setup to look at thick sections of healthy and stretched scar dermis. And in order to compare the observed differences in collagen fiber alignment and thickness, I generate color-coded maps of collagen orientation. So to quantify this, I generate Gaussian plots of orientation in degrees. And what I found was that the dispersion of orientation angles was significantly lower in the scar tissue, meaning the collagen fibers were much more aligned in stretch scars, which is actually the opposite to what we see in keloid scars. But when I looked at the thickness of collagen fibers, I saw that the thickness was significantly higher in the scar dermis, which is a feature that stretch scars and keloid scars have in common. So using the same multiphoton setup, I could also collect the autofluorescence signal coming from elastin fibers. So using the intensity of second harmonic generation images to autofluorescent images, I scored this SAA-ID index that defines the proportion of collagen to elastin fibers in the dermis. And what I saw was that the collagen to elastin ratio was significantly higher in the stretch scars. So another interesting property that was previously reported to differ during the scarring are the mechanical properties of the skin. So in order to test them, I use atomic force microscopy, and in this process, a cantilever is indented into a section of the skin, and we record the extent of deflection as the cantilever is indented. And this allows us to plot force displacement curves and fit a model that defines the stiffness of the skin. So in my study comparing stretch scars with healthy dermis, I saw significantly higher stiffness in the scar dermis. So to summarize my talk, in my study comparing stretch scars with healthy dermis, I demonstrated significantly higher collagen alignment and thickness in the scar dermis, which might have induced a change in the mechanical properties of the skin, such as dermal stiffness. In contrast, the proportion of elastin fibers was reduced in scar tissue. And we believe that this list of factors can induce a unique stretch scar environment, which can affect the cell phenotype. And that's why the next step now is to isolate dermis from stretch scars and from the healthy dermis in order to perform gene expression profiling to define the transcriptional differences between the two conditions. And as I said, this obtained knowledge will serve me as a baseline values for my patient study, which will allow me to test my hypothesis that hair follicles can induce dermal remodeling, and hopefully lead me to identify a mechanism or a cellular pathway that can then be validated using cell culture experiments. So I would like to thank my supervisor, Dr. Claire Higgins, my whole lab, especially Dr. Colin Boyle, who helped me understanding the mechanics of the skin. I would like to thank our clinical collaborators, the funding sources, and the imaging facility at Imperial College London. Thank you.
Thank you, Becca. Now it's open for discussion.
You're a boring question. Thank you for the interest. So I'm curious how you use the second harmonic to distinguish the elastin and the collagen. Is it the elastin for, like, you need additional specific staining indicate?
So no additional staining is needed because second harmonic generation imaging works specifically for collagen, which is a unique molecule, nonlinear molecule without central symmetry. So this is the only one of the unique molecules where second harmonic generation imaging can be used. So I detect the signal in a very narrow wavelength, and the rest of the signal that comes from the autofluorescence of the skin, this can show me the elastin fibers.
It could be also other tissues.
It can, yes, definitely it can be, but this is quite a well-established technique, and we believe that what I see are elastin fibers. Although it would be definitely interesting to confirm that with any other type of staining.
Are there any more questions? Thank you. Oh,
But does this scar actually affect the outcome of this transplanted hair follicle?
So this type of scars, they are actually resulted from a previous strip extraction for a hair follicle transplantation. And as far as I know, it does not really negatively affect the result, the outcome of hair transplantation. But what happens is that the clinicians reporting remodeling of the skin, they say that the skin feels nicer after the hair follicles are transplanted. So I would really want to understand why the skin feels nicer, what's the kind of mechanism that affects that.
thanks, Dr. P.
Thank you.